Welcome to EQBloom. Today we're diving into a cellular structure that doesn't get nearly enough attention, yet without it, life as we know would collapse under its own weight. I'm talking about lysosomes. Often overshadowed by mitochondria or the nucleus, lysosomes are no less vital. These membrane-bound organelles function as a cell's internal sanitation and recycling crew. They clean, break down, and repurpose a biological debris that accumulates during life's daily processes. From protecting us against invading microbes to recycling entire organelles, lysosomes are constantly working behind the scenes to maintain balance inside our cell. Over the next few minutes, we'll explore how they function, how they help defend our immune system, what happens when they fail, and why they may hold clues to aging and disease. Let's begin with the basics. Lysosomes are spherical membrane-enclosed compartments found in nearly all animal cells. Inside, they maintain a highly acidic environment around pH 4.5 to 5.0, which is perfect for the action of the hydrolytic enzymes they contain. These enzymes include proteases to break down proteins, lipases to degrade fats, nucleases to digest nucleic acids, and more. Imagine a stomach with each cell, only far more precise and self-contained. Unlike the nucleus or mitochondria, lysosomes were historically hard to study. Their transient, changeable nature made them elusive, but today we know they are absolutely central to cellular housekeeping, helping prevent the dangerous accumulation of worn-out structures and macromolecules. They originate from the Golgi apparatus, which sends them enzyme cargoes, and they're specifically numerous in immune cells, neurons, and liver cells, cells that need to manage high volumes of biological waste. Now let's talk about what lysosomes actually do. Whenever a cell engulfs external material like bacteria or viruses through endocytosis or phagocytosis, the resulting vesicle fuses with a lysosome. This fusion forms a digestive compartment where the contents are broken down. The hydrolytic enzymes break down the engulfed macromolecules into their building blocks, amino acids, fatty acids, sugars, and nucleotides. These are then recycled for reuse by the cell. This is also how cells manage internal waste, through a process called autophagy. Lysosomes digest old or malfunctioning organelles, ensuring the cell doesn't waste precious resources or energy on parts that no longer function correctly. Think of it this way: if a city never collected its garbage or recycled materials, it would soon be overwhelmed. Lysosomes are that city's waste management and recycling department, relentless, efficient, and crucial. Lysosomes aren't just janitors, they're also frontline defenders. In immune cells, like macrophages and neutrophils, lysosomes are the final destination for engulfed pathogens. Once a bacterium is swallowed by a white blood cell, it gets enclosed in a vesicle called a phagosome. This phagosome then fuses with a lysosome to form a phagolysosome, a highly acidic death chamber in the invader. But the story doesn't stop there. After digestion, some of the pathogens' remnants are displayed on the cell's surface as antigens, a critical step in alerting the adaptive immune system. Without this lysosomal function, pathogens could hide within our cells, avoiding detection. And this isn't just theoretical. Certain diseases like tuberculosis exploit lysosomal dysfunction to serve inside host cells. Lysosomes are quite literally the gatekeepers of cellular immunity. Autophagy, meaning self-eating, is one of the most fascinating processes lysosomes manage. During times of cellular stress like nutrient deprivation, infection, or oxidative damage, cells activate autophagy. Here, organelles such as damaged mitochondria, misfolded proteins, or parts of the ER are encapsulated in a double membrane vesicle called an autophagosome. This autophagosome then fuses with the lysosome. Why does this matter? Because recycling allows cells to reclaim vital components and adapt to stress. In neurons, for example, autophagy helps prevent the accumulation of toxic protein aggregates, a hallmark of diseases like Alzheimer's or Parkinson's. It's not just about survival. Autophagy promotes cellular renewal and even regulates inflammation. Without it, we wouldn't just age faster, we'd accumulate irreparable damage. Lysosomes work tirelessly, but what happens when they break down? There are over 50 known lysosomal storage disorders, LSDs, caused by inherited mutations in the genes that encode lysosomal enzymes. Without functional enzymes, macromolecules begin to accumulate inside cells, especially in the brain, liver, spleen, and bones. Take Tay-Sachs disease, for example. A defective hexa enzyme leads to the buildup of GM2 ganglioside in neurons, ultimately resulting in severe neurodegeneration. Or Gaucher's disease, when a defective glucuronoside enzyme leads to lipid accumulation in macrophages affecting organs and bones. These disorders are rare but devastating, often fatal in childhood, and they highlight just how critical lysosomes are. They're also a gateway to gene therapy research and enzyme replacement therapies, offering hope for many. Let's zoom out. Inside every cell, lysosomes are working around the clock, clearing waste, breaking down debris, recycling resources, and defending against invaders. They are the unsung heroes of cellular life. Without them, cells would swell with trash, pathogens could hide and flourish, damage would accumulate, aging would accelerate. Lysosomes are the reason cellular life remains sustainable and adaptable. As science continues to uncover the full depth of lysosomal function, from cancer prevention to longevity of research, we're reminded of a core truth: even the smallest players in biology can have the biggest impact. Subscribe to EQBloom for more science that breaks things down so you can build yourself up.